Yeah, the legacy of Gregor Mendel. You all know that one of the traits that Mendel studied was the seed color and also colors of flowers. So he chose on purpose very simple traits that can be recorded very easily, and it makes then other scientific experiments much more easy, because you don't have to focus so much on the traits, because the traits are easy to record. And also Google recognized the value of Mendel's work, as you can see in this cartoon. Maybe they got dominant and recessive not quite right, but Google recognized the importance of Gregor Mendel's work. So my task was to switch from peas to cows. Let's have a look at cows. And we are studying indeed the inheritance of coat colors in animals. And I'm a molecular geneticist, so I try to learn something about the mechanisms, how specific DNA changes really cause visible traits, inherited traits in animals. And when we look at coat color, one trait are so-called depigmentation phenotypes. And this is a work that my colleague Kurt Rögemüller initiated maybe 15 years ago, in a time when there was no next generation sequencing yet, so it was in the early times. And you see these beautiful patterns on these Swiss cows. It's a trait that has an intermediate mode of inheritance, or nowadays we call it semi-dominant. So the heterozygous animals have this spotted pattern on the head, which in Switzerland is called Blüm, and in the English-speaking worlds this is called color-sided. The cow on the right is a homozygous animal which has a more severe phenotype, a more pronounced phenotype. And at the time we mapped the gene, so with the markers, as you just heard in the previous talk. And this gene causing this specific pattern in these cows mapped to chromosome 6, close to a gene that's called KIT. And KIT is a gene that is very well known in mammalian biology. It mediates a signal that is required for the survival of melanocytes, the pigment-producing cells. And this is a very important gene, and it needs to be very, very tightly regulated. If there is too much of KIT expression, you may end up with tumors because cells proliferate too much. And if there is too little of KIT activity, one of the cell types that will die are the melanocytes, and you end up with having unpigmented white spots on your skin. So we mapped this, and this mapped to chromosome 6. We sequenced the coding parts of the gene, but at that time, we were not able to sequence really large scale, and there was no coding change in the KIT gene. So we kind of paused at that point. And then at a scientific meeting, Michel Georges from Liège approached me, and he got in touch with me. We started talking, and he said, yeah, he's also studying this trait, color-sided, in a different breed of animals in the Belgian blue breed. And they also mapped the gene causing this color, and in their population, it mapped to chromosome 29. And phenotypically, it looked very similar. So this was a puzzle to us. And Michel Georges and Carole Charlier and their postdoc Keith Durkin, they really pursued this and followed this up and did a lot of very interesting and very cool work that I'm going to present very briefly. So with their marker studies, when you look at this picture, without going into details, their strongest signal was on chromosome 29, but they also saw something on chromosome 6 where we had our signal. And then they looked deeper into their marker data and produced this graph, which shows that markers that are supposedly on chromosome 6, close to the KIT gene, gain in intensity. So basically, this tells you that a piece of DNA from chromosome 6 has been duplicated. An additional copy of a relatively large chromosomal segment is present in these cows. And when they did cytogenetic experiments with a probe against the KIT gene, they realized a segment of chromosome 6 has duplicated and translocated to chromosome 29 in the Belgian cows. And to make a very long story very short and simple. It was not that simple at the time. They sequenced a lot of animals, and in the end, they recognized what must have happened. And on this picture, you have to start at the top right. Start at the top right. That's a wild-type chromosome 6 of normal pigmented cows with the KIT gene. And what must have happened in breed history is that a piece of this chromosome 6 must have excised, formed a circle. The circle must have opened at a different position, and then this piece of DNA has integrated into a piece of chromosome 29. And that's what you see at the bottom left. That would be the chromosome as you see it in Belgian blue color-sided cattle that have an additional copy of KIT on chromosome 29. But that's not what happened in the Swiss cows. In the Swiss cows, yet another translocation has happened. A part of the chromosome 29 segment excised again, formed again a circle, and reinserted to chromosome 6. So the Swiss cows have a short piece of chromosome 29 on their chromosome 6, which gives rise to two different alleles that are phenotypically, have very similar consequences. Phenotypically, it's impossible to distinguish these animals, and the functional effect is a misregulation of KIT. These animals produce a little bit less KIT, and that results in this very striking phenotype. And Hans Sölkner and many other researchers helped us to collect animals across the globe. These must be very old alleles that must have arisen very early on in breeding history. These phenotypes are really found in many diverse cattle breeds, and they have even been integrated into yaks, which is another species. So you find animals with these phenotypes across the globe. Let's switch to another phenotype, looks very similar. The beautiful Pinskower cow. This is a project that Kurt Rügemüller and his doctoral student Lucia Küttel started a few years ago. Now much more modern times, 10 years later, we have all these advances in sequencing technology. Nowadays, it's no longer very difficult to sequence an entire genome. And when they do this, they find an allele that is a little bit similar to what we saw in the color-sided animals. So this cartoon, it's not important that you focus very much on the details. However, what has happened here is that a very large piece from chromosome 4, a 300 kilobase pair piece from chromosome 4, has also duplicated and integrated right behind the KIT gene on chromosome 6. So we have another example where a huge chunk of genome duplicated, inserted, and when these pieces of genome insert close to the KIT gene without interrupting the coding sequence of the KIT gene, there is a good chance that this will influence the regulation of KIT and will then lead to white spotting phenotypes of some sort. Now there are more of these white spotting phenotypes out there, and another phenotype that was of interest to me is the belted phenotype. And there are brown cattle that have this white belt around the midsection. And this is work that we performed with Kurt Rügemüller and my PhD student Nivedita. And all of these cows with these very special colors, they are very dear to the breeders. Breeders value them. I would say it's possibly more for an emotional aspect than true performance that you can measure in any performance trait. But supposedly they bring luck to the herd. And you will find them in art, in not so recent art, but also in older pieces of work. So for this phenotype, we are sure that it's at least 700 years old. There is a painting not far from here in Klosterneuburg, which is dated to the early 15th century. And it's a little bit hard to say, but I would say this oxen is a belted oxen in the painting. Today, we know of 3 breeds where this trait segregates. The brown cattle, which is an alpine breed, and then if you go northwest, the Lakenfelder from the Netherlands, and if you go further northwest, the Galloways from Ireland. The belted Galloways also carry this trait. We mapped again with the markers, and this time we ended up not at the KIT gene, which was maybe our first candidate gene. This time we mapped it to a region upstream of the TWIS2 gene, which at that time was not known to have anything to do with pigmentation. The function of TWIS2 is not really clear. It's a transcription factor expressed during development. And genome sequencing again revealed we have a relatively large structural variant. In this case, it's a 6 kb sequence upstream of TWIS2 that is quadruplicated. So in the belted animals, we have 4 copies. And our hypothesis was that this could possibly lead to a misexpression of TWIS2, and which then might again have an effect on the development of melanocytes, so that the melanocytes do not properly develop in the midsection of the animals. And because the trait is inherited as a dominant trait that would indicate gain of function, an additional function, our hypothesis was that TWIS2 is probably upregulated in these animals. Now, how do we prove this? This is maybe different from the way that Mendel did his research. When we want to start to investigate the development of early melanocytes, there is the model organism for this type of experiments, which is the zebrafish. Zebrafish embryos are very small, and they are transparent in the first days of their life. And when melanocytes appear, you can see them visually through the microscope, appearing as dark spots in the otherwise transparent embryo. And we produced embryos that overexpressed twist, and those embryos that overexpressed TWIS2 had statistically significant, but not very striking. They had less melanocytes than the wild-type counterparts. And we took this as a supporting evidence that indeed this non-coding structural variation that we found in the belted cows leads to the upregulation of TWIS2 and causes the belt. Now I would like to switch species. Let's move from cows to goats. Goats are very important in Switzerland, and here you see Valyrian goats that also have a depigmentation phenotype, so they have no melanocytes in their back half. And this is a trait that I'm super interested in, but today I cannot yet tell you the solution. I don't know what's causing this. This is now work done by my PhD student Jan Henkel and Christine Fluri from the Bern University of Applied Sciences. And in the course of this work, we studied several goat breeds and compared them. And in Switzerland, there are many goat breeds. I think very beautiful animals with very striking patterns. Now we have to consider a new mechanism. I won't go into details, but these goats, they all have a complete set of melanocytes. So their entire skin is populated with melanocytes, and the patterns that you see on these goats are caused by changes in the so-called pigment type. So melanocytes produce melanin, and mammals are able to produce 2 different types of melanin. The so-called eumelanin, which is black or dark brown, and the so-called pheomelanin, which can vary from dark red, yellow, to almost white. And these white goats that you see here, the Appenzell goat and the Saanen goat, they express a very, very light pheomelanin, but they have pigment cells. And it is known that this so-called pigment type switching is controlled by a gene called agouti. And when agouti is expressed, the goat will, or the melanocyte will produce pheomelanin, and when agouti is not expressed, the melanocyte will express the black eumelanin. This is all not new. In the 90s, Adelsteinsson, an Icelandic scientist, did a cross with different goat breeds, produced a huge herd, more than 200, 300 animals, counted very precisely the phenotypes, just in the same way as Mendel counted the phenotypes of the peas. And from this work, Adelsteinsson postulated that there should be 11 different agouti alleles in goats. That's a huge number of alleles for a single locus in a mammalian species. but there's also a huge number of patterns in goats. So we tried to get into the molecular genetics of these sequenced genomes of goats and could solve at least some of these patterns. And we have again these large structural variants in the genome where huge sections are amplified. So the two white goat breeds, the Appenzell goat and the Saanen goat, their color is, or their pattern is due to the same allele. And this is a triplication of a 150 kilobase per section that contains the entire coding sequence of the agouti gene. So they basically just have more agouti, and if it's higher expressed, it's not a huge surprise that they produce only pheomelanin. Then we have the Bündner Strahlenziege that has a short piece of DNA in 8 copies, and this piece is in front of the gene in the promoter region and changes the expression and makes this striking pattern. And I ask you to really focus on the faces of these goats. They have 2 white stripes where only pheomelanin is produced. And then the next pattern, these are the gemsfarbige Gebirgsziege and St. Gallen Stiefelgeiß, have yet another copy number variant, and look again in their face. They now have stripes in the face at exactly the same position as the others, but now they produce only eumelanin. So the regional expression has just switched just the opposite. I find this quite fascinating. And then the last one are the Peacock Goats. That's an allele that Adlersteinson did not have in his cross, so there's yet another agouti allele. A very complex pattern, and when you look at the breakpoints of the amplification in the genome, you can see that it's a derivative of the allele that we saw before. We followed this up. Is it all true that this is due to differential agouti expression? We took skin samples from these goats, measured RNA expression, and indeed, this confirmed the white and the red pigmentation is due to high agouti expression. And our RNA experiments also revealed that in goats, the agouti gene has at least 3 different promoters and a large number of untranslated exons. So the coding sequence of agouti is conserved across all mammals, but the 5' region that is controlling the regional expression and gives you defined patterns, this is not so super well conserved across mammals, and it's very complex in goats. Now I would like to come back to my Valyrian goats. I still don't know why they are not pigmented in their back half, but there are 4 coat color varieties. And there were like rumors across the breeders, these are super rare subpopulations, they are possibly unique breeds, we need to conserve them. Yeah, when we had our data, we looked a little bit closer into this, and unfortunately we think they are not super unique, these 4 breeds. What you can see here is a mixture. With our knowledge of the agouti alleles, we could trace this. So the most common variety, and today the modern breed standard, is the so-called blackneck goat. That's the official breed standard, and of those, many animals exist. And the other 3 are rare varieties, but the rare varieties are due to integration of the pattern alleles. The white Capra sempione is due to an integration of the Saanen allele. The Grüne Nochte is due to an integration of the Peacock allele, which is hard to see because the Valyrian goats have these long hair and you cannot see the pattern so well. But if you do it on the molecular angle, then you can easily verify that it is the Peacock allele. And the Copperneck goat, that's a brown color variety, is due to an integration of a mutant to one allele that came from Toggenburg goats. We could a little bit clarify the breed history and the origin of these varieties. And then for the last couple of minutes, I would like to talk about the color of dogs and wolves. You saw this very nice picture from Professor Pockenberger before. This is work that has been done mostly by Danica Banish from the University of Davis, who spent a sabbatical in Bern, and also Chris Kalin and Greg Barsh, who helped us with evolutionary aspects. So when we look at wolves, there are actually 3 different color morphs existing. This is all the same species. The gray wolf that you all know from Europe. Then there is a very light variety of wolves living in the Himalaya, and there are the completely white Arctic wolves in the American Arctic. We started our work with dogs, and in dogs, a number of patterns is historically known. And on the right side, you see the old traditional allele designations. The top 2 rows were called fawn or dominant yellow. Then we have agouti in the 3rd line. In the 4th line, this was called saddle tan, and the bottom line was called black and tan. And in the literature, there were publications that reported very small changes that were claimed to be causative or that were very well associated, but these publications didn't read completely right, and diagnostic laboratories who do commercial genetic testing for dog breeders reported that some of these markers didn't work so well and that they had a relatively large fraction of results that could not be really explained. And this was then also summarized in another publication, which even suggested a novel chromosomal rearrangement. So the coat color scientists in the dog world, they knew something is not right there. Our picture of things is not right. And then Danica came to Bern, and the first thing she did is she said we need to do away with these old color designations. They are not appropriate. Let's replace them by new ones and introduce another allele. So the top 2 rows, although these phenotypes are similar, they are not identical, sometimes very hard to distinguish in reality, but she distinguished dominant yellow, which is a completely yellow dog, from shaded yellow, which is a dog that has very subtle black tips of the hairs. And what we did, or what we had at the time, we had a lot of genome sequences from all our work in dog genetics. So we had hundreds of sequenced dogs with different colors. We had photographs of these dogs, and we could correlate genomic sequence with phenotype. We also had whole transcriptome data because we are studying inherited skin diseases where we sequence RNA from skin samples from dogs to learn more about changes in gene expression in diseased skin. And with the transcriptome sequence data, we characterized in more detail the genomic locus. So the agouti gene is the ACYP gene. And like in all other mammalian species, it has 3 coding exons. In dogs, it has 3 promoters. And the first promoter, we still don't know the exact function. The second promoter is something that we call ventral promoter that drives expression on the belly of the dog. And the third promoter is the so-called hair cycle-specific promoter that regulates the expression of agouti during the growth of a hair. If you pull the hair from a wolf, it will be banded. So the tip will be black, in the middle it will be yellow, and at the base it will be black again. This is so-called agouti phenotype, and this is the function of the hair cycle-specific promoter. And when we look at the genome sequences of the different color patterns, we find many different alleles. I apologize, this is now a busy and complex slide. So at the ventral promoter, we saw 2 different alleles, and the difference between these alleles is, again, non-coding structural variation, but now we are not talking about many thousands of base pairs. Now we are talking about the absence or presence of a sign element. A sign element is a short transposable element in the mammalian DNA. It multiplies via an RNA intermediate, and that's the reason why it's called a retroposon. And we have two alleles, one that we call VP1, ventral promoter 1, and the other that we call VP2. And VP1 encodes an overactive promoter. That's more active than the wild-type promoter if we consider the European grey wolf the wild-type situation. So the agouti pattern is what we consider the wild-type situation. And VP1 is an overactive promoter. And at the hair cycle promoter, we found 5 different alleles. HCP1 is again an overactive promoter that gives rise to more expression of agouti. HCP2 would be the wild-type situation in the agouti dogs. And HCP3, 4, and 5 are all loss of function promoters where the function is completely lost. And the different patterns that we see in dogs can be easily explained if you consider that the expression of agouti is controlled by more than one promoter. So you have to consider the combinations of the promoter. And at the bottom, you see these 5 different patterns, and the arrows indicate the activity of the promoters. So when both promoters are overactive, you end up with a dog that produces so much agouti that the entire pigmentation is pheomelanin, and the dog is either yellow or white or red. In the shaded yellow dog, we have a normal activity of the ventral promoter, and that means that the hairs on the back get already the black tips. Agouti is the wild-type situation where both promoters have their normal activity. Black saddle, you have an overactive ventral promoter, which gives you a completely yellow belly. And because the hair cycle promoter is inactive, there is no banding of the hairs on the back, and then the hairs on the back are completely black. And black back, the regular ventral promoter and the loss of the hair cycle promoter. So if you understand the combinations, it's, I think, fairly plausible how the different patterns arise. And at that time, our colleagues Greg Barsh and Chris Kalin had a closer look at the evolutionary history of these alleles. And what you see here is a comparison of a larger region, and at every nucleotide difference that we saw, you can have either yellow if the allele corresponds to the genome reference, or blue if it's the alternate allele. And now you have to remember, or it's a little bit peculiar, our genome reference comes from a dominant yellow boxer. So that's possibly not the wild-type allele, but dominant yellow is a very common color across dogs, and therefore this is the allele that's represented in the genome reference. And the first thing that you may notice is in the top line we have these white Arctic grey wolves, and they look virtually identical to dominant yellow dogs. So it's actually the same allele that's causing yellow color in dogs and the white color in the North American Arctic wolves. And when you look a little bit further, we have this 5' part, and Chris Kalin took a 48 kb section, and when you take the markers there and do a phylogenetic tree, you come up with the phylogenetic tree that you would expect. So dogs and wolves cluster together, and there are the other canid species, and you get the normal phylogenetic tree that is very well known. However, when you look at the hair cycle promoter, when you look at this picture here, you see that we have the top line that are completely yellow, and then the bottom 3 or 4 lines that are completely blue. So a huge difference between these two. And actually, this DNA, when you look at the sequence, it really doesn't look at as if it comes from the same species. There's just too much difference. And when you take this piece of DNA and do another phylogenetic tree, all of a sudden, the dogs don't cluster together. The gray wolves and the agouti, black saddle, black back dogs, they cluster together where they should be in the phylogenetic tree. But the dominant yellow dogs and the Arctic wolves, they cluster somewhere completely else. You see that they are now farther away from dogs than, for example, the coyote or the golden jackal, which are different species. And this analysis, to us, there is only one conclusion that can be drawn, that this piece of DNA that's causing the dominant yellow pattern in dogs and the white color in Arctic wolves has been introgressed from another species. From a species that's now extinct, it's no longer there. We looked at all known canid species that are around. And when you reconstruct the history, this introgression must have happened at least 2 million years ago. So there must have been a canid that's now extinct that must have crossed with early wolves. And possibly this led to a better adaptation during the ice ages. When there are light environments with lots of snow, then it may be an advantage if you are very light. And by this means, the HCP1 promoter has been introgressed into wolves and now also in modern dogs. So to summarize, this agouti gene and the patterns are caused by modular promoters. We have independent hair cycle specific and ventral expression. And when you want to understand the patterns, you need to consider the combination of the two. There are 7 haplotypes that explain 5 different patterns. And the VP1 allele is already more than 50,000 years old. That can be detected in ancient DNA samples from ancient dogs and wolves. But the HCP1 allele is really fascinating because it's derived from a species that's no longer on the planet. And we still see like a remnant of this old extinct canid species. So the fascination of Mendel for the inheritance of coat color is still alive. I'm completely fascinated by the inheritance of coat colors. And although it may seem simple, there are sometimes some complex mechanisms behind. And with that, I would like to thank all the many, many collaborators who helped to perform this work. And I thank you for your attention.
This video was supported by the project number ATCZ278 with the title Gregor Johann Mendel's Legacy to Science, Culture and Humanity, which is co-financed by the Interreg Austria and Czech Republic. Down below you see the title of the project in German and Czech. Here I would like to take a brief moment to explain what the project is about, because it is actually an awesome one. The main goal of the project is actually to promote Mendel's legacy by long-term cross-border collaboration in science and culture. There is a strategic plan developed that will promote this legacy in the fields of science and culture in Czech Republic, Austria, but also worldwide. This promotion will take various forms, be it meetings, workshops, publications, audio and video materials. One of the main parts of the project is actually rebuilding Mendel's original greenhouse in the Augustinian Abbey in Brno. This newly rebuilt greenhouse will be a new landmark, not just the city of Brno or the country of Czech Republic, but the entirety of the scientific fields of genetics as such. Mendel's greenhouse is the very place where genetics was born, with the first genetic experiment conducted on peas. Therefore, it is a really, really important location for the field of genetics. As a major outcome of this Interreg project, this greenhouse will become available for the general public, but also the scientific community as a multi-purpose gathering place for cultural and scientific events. This and much more you can find out if you visit the website gjm200.cz, which of course stands for Gregor Johann Mendel, and the 200 indicating the 200th birthday of Gregor Mendel, which we celebrate in 2022. Once again, a great thank you to the project and Interreg for the support of this video. Thank you for your time you spent on it watching. And from my side, I just wish you a very nice day.